Hello class, this is our lecture for our biochemistry class, namely entitled Enzymes. Now in this lecture, we will discuss the definition, compositions, classifications, functions, and enzyme activity regulation. So this will only be the scope of our lecture for today since this is just an introduction. And we will discuss further the enzyme kinetics, the mechanism and equation in the latter part of our lecture, which is the supposed to be covered in the part 2, but this is lecture again is just an introduction. Okay, ngayon, dito iikot guys yung ating discussions here. Dito, pag ganyan. Ito, dito siya iikot. Ngayon, unahin natin from definitions, and then pang fourth yung functions, pang fifth yung enzyme activity regulation. So, unahin natin yung definition. So, what are enzymes? Or what is enzymes is our subject. The enzymes are proteins. Wow, it is a proteins. Noong bata ako, akala ko dati, sure ko lang, is yung mga proteins natin, it's just made up of muscles and their basic component is amino acids. Pero hindi pala ganun. So the proteins has a different types of proteins based on their classifications. At isa nga dito yung kanyang biological function. And ang nangunguna doon, guys, is yung ating enzymes. So ang enzymes natin is a tertiary and quaternary structure of our proteins. It is a catalyst, a biological catalyst. So when we say biological, involves life processes that accelerates a biochemical reaction and catalyze almost all reaction within the cells or pwede rin outside the cells. And after that, it is not permanent change in the process to the enzyme substrate activity process that we will discuss later. So that is the definition of enzymes. Now, we will go on to our composition. So, ano ba yung enzymes natin? Di ba na-discuss naman kanina that it is a tertiary and quaternary structure of proteins. Pero, as we all know, na malalaman nyo pa lang ngayon, it is composed of a coenzyme or metal ion na covalently bounded to the apoenzyme or apoprotein. And ang tawag natin doon, guys, is yung ating prosthetic group, the coenzyme or metal ion part of our enzymes. And then yung protein part, the enzyme naman natin is called the apoenzyme and apoprotein. So is the coenzyme or metal ions and then apoenzyme combined together, yun yung enzymes natin. So sir, ano po ba yung coenzyme and metal ions na yan? Okay, yung mga metal ions natin na yan, as discussed, syempre sa general chemistry natin, it is the copper ions or any other ions that present in the enzyme as a para mag-function totally yung enzymes natin. And then, doon naman sa ating mga coenzymes or yung ating mga cofactors, ayan, when we say coenzyme, guys, di ba? Kasi evident naman na that a metal ion is just a cation of metals. So, ano naman yung enzyme? It is a complex organic compounds bounded to our enzyme to function efficiently so that the enzymes can function efficiently. Okay? And ano-ano ba yung mga yan? Okay. Ang common example, guys, ng ating enzymes is yung ating iron 2 positive kung sa metal natin doon sa ating hemoglobin. Sa may atin namang coenzymes yung mga porphyrins natin doon sa ating hemoglobins or comprise the heme group doon sa ating bloods in our red blood cells na composed nga ng hemoglobin that which is giving it a red pigment and so on and so forth. And then sa ating enzymes natin, actually this one is covered by the function. So paano ba, how does it catalyze a chemical reaction? So paano ba nangyayari yun? Kasi nag-act siya as catalyst. Siyempre, we all know the definition of catalyst. Participates in the reaction pero hindi siya nakukuha as a product. Ganito yung mangyayari dyan. They catalyze yung substrate natin or the reactants, okay? The reactants will go on the active site part, which we will discuss in the latter part of this lecture dito sa active site ng ating enzymes to proceed a certain chemical reaction, either oxidation reduction or rearrangement kung ano mang process yung magaganap dyan or redox reaction na yan. So it will go here at the active site and then ika-catalyze na ng enzyme yan via some sort of complex chemical reactions to form a product. And then ang enzymes natin has a suffix of ASE. Suppose that you want to catalyze the reaction of sucrose, which is our table sugar, So, sucrase yung pangalan ng enzymes natin yan. So, matatakal yan doon sa may specificity. And then when we say we want to catalyze or the hydrolysis of our lactose, the enzyme responsible for the hydrolysis of lactose, let's suppose it's an hydrolysis enzyme, it is a lactase. And then third naman natin is the maltase if we want to catalyze the hydrolysis of our maltose. Now, punta naman tayo sa kanyang classifications kasi nanggaling tayo sa kanyang compositions. The classification of enzymes here, ayan, we have the class number 1 to 6. And I want to highlight that the arrangement or the order of this classification number matters. Kasi meron tayong EC1, EC2, EC3, EC4, EC5, and EC6. What is EC? On yun, ang meaning nun is just letter E, letter N, letter C, which is, meaning na acronym nun is a enzyme classification. So for our class number 1, we have the oxidoreductase for the redox reaction enzymes. So the enzyme, ang mga ating oxidoreductases enzymes catalyzes the oxidation and reduction process. And then for our class number 2, we have the transferases catalyzes the group transfer reactions. For the hydrolases enzymes, catalyzes the hydrolysis reactions. For the lyases, okay, the word lyas, okay, it is the removal of groups or the formation of the whole bond. Yun yung reactions na ina-catalyze niya. For the isomerases, it is a transfer of groups within molecules to yield isomeric forms or catalyzing the isomerism of our substrates. And then the ligases, formation of the mga bonding or the condensation reaction, such as the condensation reaction of our fructose and glucose to yield a sucrose. Ayan. Ayan. So those are the classifications. Ayan. So EC, okay, I want to corrections doon na it is a enzyme commission number system. So EC1, EC2, EC3. Okay, that's enzyme classification. Okay. And then, punta naman tayo sa functions natin. As discussed earlier naman kanina, na yung functions ng enzymes natin catalyzes the reactions by the substrate entering to the active sites of our enzymes. So, ganito ang nangyayari dyan. So, how does it catalyzes the reaction? Ang nangyayari kasi doon, guys, is lowering the activation energy of our reaction. So, what is the definition of activation energy? Okay. Suppose we have these two graphs here. This one here is the catalyzed reaction, and this one here is the uncatalyzed reaction for the red line. Ang gagawin ng enzymes natin is to lower the activation energy. Sir, what is the activation energy? The energy required so that the chemical reaction proceeds. Okay? Kung mapapansin nyo, parang ang analogy lang yan, guys, is gusto mong magpadulas sa may bundok. Wala kasi yung illustration dito. Ayan. Pero mababa lang yung inakyat mo para makapagpadulas. Those are the enzyme catalyzed reaction. Pero dun sa may non-catalyzed reactions, maalayo pa yung akyatin mo. Mas mataas na hamak pa ang akyatin mo dun sa may tuktok ng bundok bago ka makapagpadulas. Which is evident naman na mas napagod ka dun sa may pag-akyat, dun sa may mas mataas na bundok. Kasi ang goal mo lang naman is makapagpadulas. So, ganun yung ating activation energy, the enzyme catalyzed reaction. Pagka nagka-enzyme ka, halimbawa, mas mababa akatin mo bundok para makapagpadulas. So, for the reactions to proceed. So, that's it. And ang mangyayari dyan, as discussed earlier, yan nga so free energy, lower energy activation energy required so that the progress of the reaction proceeds. So, mas mapapabilis yung reactions natin. And ang common example nito, guys, is yung ang hydrolysis or the decomposition of hydrogen peroxide into water vapor and oxygen. So, paano ba nangyayari yan? Suppose that we have two test tubes containing hydrogen peroxide. And then, this one here is the uncatalyzed. This one is catalyzed. Okay? And then, the enzyme responsible for the decomposition of hydrogen peroxide is the catalase enzyme, di ba? ASE. Ngayon, kung mapapansin niyo, after pouring the liver homogenate to the second test tube, which is our catalyzed reactions or the decomposition of hydrogen peroxide, is nag-yield na agad siya ng water vapor and bubbles. Unlike dito sa uncatalyzed, is walang nangyayari. So, in a matter of seconds, decomposition occurs. So, kailangan ninyo talaga ng enzymes para mas mabilis ang reaction. Ganyan yung nangyayari sa katawan natin. It is a series of enzyme catalyzed reactions so that we can function to our everyday life. Okay. And if kung na-try ninyo guys, yung natin, kung gusto nyo matry to in real life, kunwari nasugatan kayo, and you want to observe the decomposition, immediate decomposition reaction of hydrogen peroxide, nasugatan kayo, tignan nyo ninyo yung difference. Let's say may sugat ako dito sa may part na to. Mag-pour kayo ng hydrogen peroxide sa walang sugat, doon sa may sugat. Magkakaroon ng bubbles doon sa may sugat. Kasi nga, our wounds or yung sa ating from the veins natin, blood natin contains a catalase enzyme. So, ganun yung mangyayari. So, if you want to test, okay, for your own curiosity, pwede nyo subukan yun. Okay? Ayan. So, again, ano ba nangyari doon? Suppose that this one here is the catalase enzyme of the hydrogen, and this one here is the hydrogen peroxide. Ganito kasi ang functions niyan. Okay? Kunin natin. Let's have a here. Okay. Ayan. Suppose here that this one is yung ating H2O2. Ayan, ito yung ating H2O2, and this one here is the catalase enzyme. Catalase enzyme. Ngayon, ang mangyayari dito, this one here is the substrate as S, and then this one is substrate as E. Ang mangyayari dito, guys, is we have the enzyme plus substrate. So, ang mangyayari, magjo-join sila to form a, ayan na, this is not the exact drawing, pero sana ito yun. Okay? This is the enzyme, this is the substrate, forming the enzyme substrate complex. So this one is the ES. Then after that, the hydrogen, di ba? The substrate, which is the H2O2, decomposes into H2O and oxygen atom. So mangyari dito, we have this. Siyempre, di ba? From one product, naging two. So we have two molecules here. Ayan. So mangyari ngayon dyan, this one here is the enzyme product complex. Ayan. And then after that, so mangyayari dito is aalis na yung ating H2O2, ay H2O and oxygen. So ayan, and this one here, the enzymes. Yan, so this one's the E, the catalyst. Dito na ngayon yung ating H2O plus ating oxygen, and this one here is the enzyme, or also known as the products. Ayan, plus product. So ito yung ating enzyme catalyzed reaction. Illustration. This is the reaction. Okay? Ayan. So, it is a reversible reaction na mangyayari. Ayan. This is the illustration. Ganyan yung nangyayari through the enzyme catalyzed reactions. And then, after that, ayan pala, may illustration pala nakalagay doon. Ayan, magjo-joins to form a enzyme substrate complex. And then, maghiwalay na sila to form the product. Ayan. So, ang tawag doon sa ating dito sa may part ng enzymes natin na ito is yung ating active site. Active site na kung saan doon nagjo-join yung substrate to the restricted region of the enzyme para mag-bind doon so that the reaction can proceed. So the active site, enzymes, and substrate, okay, to undergo a enzyme catalyzed reactions. And then the active site natin, ito yung mga characteristics niya. When you say active site, this is the part of these proteins where the substrate binds to the enzyme. So, babasahin ko na lang. Demonstrates specificity. So, specific lang yan. It's just like a lock and key model. For every lock, there is a specific key para mabuksan, para ma-unlock ninyo yung pintong yon. So, that's the specificity. Hindi sige mamaya discuss. Relatively small, three-dimensional region with the enzyme, and substrates are initially held in place at the active site by weak non-covalent reversible interaction. So it is held by weak non-covalent reversible interactions for the enzyme and substrate complex. Ayan. Ngayon, pupunta tayo sa may specificity, sinasabi ko kanina, that yung, kunwari That's the one enzyme can catalyze four substrate, di ba? Diniscuss ko kanina that the sucrase enzymes catalyzes the sucrose, not the lactose. Kasi nga, our enzymes exhibits a specificity. Na kung saan, meron tayong, I think, 4 types. So, meron tayong absolute specificity for the catalysis only one reactions. And then group specificity, enzyme that only reacts with the specific functional groups. So, what are the functional groups ng enzymes natin? Ay mga organic compounds. Siyempre, napag-aralan niya sa akin yung organic chemistry such as the carboxylic acids, esters, and amines, and so on and so forth. So that's the group specificity. And then meron din tayong linkage specificity. Okay? Ang kina-catalyze lang niya is yung particular type of chemical bond such as the either ionic bonding or what are the other types of bonding? Basta, covalent bonding or non-covalent bonding. Okay? Depende doon sa may molecular structure ng ika-catalyst ng reaction. And we also have the stereochemical specificity. Sir, what is stereochemical? Yan yung mga R and S configurations natin. Some enzymes catalyzes the R configuration or ng ating organic compounds, but not the S. Okay? So what are the R and S configurations? That's the rest dun sa may inyong organic chemistry na yon, and that is stereochemistry. Okay? So ayan, this is just to reiterate that the specificity of the enzymes. The sucrase will only catalyze the sucrose but not the lactose and maltose. The lactase will only catalyze the lactose molecule but not the sucrose and maltose. And the maltase is only catalyzes only maltose but not sucrose and lactose. So that's the exhibition, specificity of our enzymes related to our enzyme substrate composition. So ayan, and we also have induced fit model for the specificity of enzymes. Natatandaan ko nung sinabi ng teacher ko na si Sir Feliciano na kung ang enzymes natin ay napaka-specific or only one substrate, napakadaming enzymes sa katawan natin. And I think he said that this is the best model that we have to look at. Kasi, ang principle kasi nito, once na kunwari, active site and substrate, okay, nag-fit, go, magpo-proceed na ng reactions. Pero kapag, let's say, sucrose or the sucrase, so isa-isa. So, sobrang daming enzymes. And I'm still working on it I'm still trying to verify that. Working on it, studying on it, kasi hindi natin alam kung ano ba talaga ang specific reactions ng ating induced fit model. So, ayan. So, induced fit model, basta nagkasya, okay, configurations, go, reactions, proceed agad. Doon na maokan natin yung kasing describe ko na the first specificity, specifically yon sa mga ating lock and key model. Isang susi, kada lock. Dito naman, kahit ano. Okay? Ganun. So, done na tayo dyan. So, next naman natin is the factors affecting the enzyme activity. Factors. Kasi, yung ating natin, di ba? For us, ang analogy nito is, di ba tayo, some of us functioning well dun sa may hot environment. Some of us is nagpa-function naman dun sa may ambient environment. Yung iba naman is sa may mga malalamig. Di ba? So, kapag nag-exam ka, saan mo mas gusto? Dun sa may mas mainit na room or tamang temperaturang room? Siyempre, sa akin, ako lang naman, is makapag-function ako ng maigi kapag nag-exam ako dun sa may tamang temperaturang kwarto. Kasi kapag mainit is, siyempre, distraction yon. So, ganun din sa enzymes natin. Meron din silang specific environment so that they can function efficiently. And yun yung mga factors. We have to consider those factors, okay? Ang factors na yun is, ayan, we have the environmental conditions, cofactors and coenzymes, and enzyme inhibitors. So those are the 3 factors affecting enzyme activity. Okay? So next natin is analogy natin dito, okay? Siyempre, temperature. Kapag sobrang init, our enzymes tend to denature, thus losing their functions as a catalyst. So hindi na nila mape-perform yung function nila kasi nga na-denature na nila due to the extreme temperature or sobrang init. Siyempre kahit sino naman, di ba? Ayun. So pag nag-exam ka, that is yung sobrang init. So enzymes must function at a specific ambient temperature so that they can function efficiently. Ayan. The next natin is yung pH. 'Di ba? Umiinom naman tayo, 'di ba? Pag umiinom tayo ng tubig is neutral, minsan naman basic. Pero depende. Pero sa enzymes natin is nakaka-affect yung pH. Okay? Ang kuha natin dito, ang efficient pH na nakapag-function yung ating enzymes natin is between 6 to 8 to reach the highest peak. 4 to 10, ayun, medyo depende na rin yun. Depende rin sa enzymes, okay? So, ang point ko dito is dumidepende din sa pH and the ionic concentration or the salt ions sa kanyang environment. Okay? Next natin is the cofactors and coenzymes. Okay, such as the inorganic substance, di ba? Yung cofactors natin, such as zinc and iron and vitamins, respectively, are sometimes need for proper enzymatic activity. Parang yan sa analogy natin is kailangan ng tubig para makapag-function ka everyday. So, ang binanggit ko kanina, yung ating example natin dito is yung ating iron. Yung iron natin must be present in the quaternary structure of hemoglobin so that makapag-bind siya more efficiently ng oxygen atom para ma-deliver ng hemoglobin natin through the works of iron, yung oxygen dun sa may katawan natin so that we can function efficiently. And we are all familiar that lack of iron, di ba? So it is that term anemic na kina natin. Na kapag lack of iron, syempre, enzymes, wala kang iron, iron yung enzymes mo kasi hindi ka nag-intake. Ang tendency nun is headache, fatigue, and feeling of tiredness ang mararamdaman mo, di ba? So, hindi lang ang enzymes ng katawan mo ay apektado rin ng buong katawan mo. So, as you can see, we are all connected from the atoms in our body to the biosphere to the organism. So, ganun lang naman. So, some kind of weird stuffs. And then the other is the mga ating coenzymes, di ba? Kanina is cofactors. The coenzymes, these are these. The coenzymes for every vitamin, such as ayan, no? And this is our the reactions, the mediated, the catalyzing, as I can say. Ayan. So, coenzymes for the vitamin biotin, flavin coenzymes for the riboflavin. So, ayan. So, those are the coenzymes for every vitamins. Okay? So, kapag natin, ayan. And then, next naman natin is yung ating enzyme inhibitors. Okay? So, what are the enzyme inhibitors? Or ito yung mga panggulo sa enzymes, sa mga mang-aagaw sa substrate. So, ano-ano ba yun? We have competitive inhibitor, we have non-competitive inhibitor, and we have uncompetitive inhibitor. So, the two examples of enzyme inhibitor is the competitive inhibitor. So, sila yung panggulo dun sa may substrate na yun. So, ito yung mga chemicals, okay, babasahin ko lang. Chemicals that resemble an enzyme's normal substrate and compete it with the active site. So, nag-aagawan sila dun sa active site. So, this is one is the competitive inhibitor. Inaagawan ka na ng site doon. Instead na ikaw yung pumasok doon, iba yung inhibitor yung pumasok. Yun yung competitive inhibitor natin. Doon naman sa may non-competitive inhibitor, binabackstab niya yung enzymes natin, di ba? Kung ito, direct, natin, kung ito, direkta ang inaatake, dito, inaatake ka, patalikod. But not directly to you, but to the enzymes itself para hindi ma-catalyze yung substrate or the reaction mo. So, ang ginagawa niya, umatake siya dito sa likod ng enzyme. So, ang mangyayari nun, syempre, ma-alter yung shape ng active site natin. And ang tendency nun is, hindi na mag-fit yung substrate dun sa may, hindi na mag-fit yung substrate dun sa may enzyme. So, it is altered na. So, makakapag-proceed ka pa ba? Yes, makakapag-proceed pa ng reactions yung substrate natin, but not efficiently because the non-competitive inhibitor alters the shape of the active site of the enzymes. Okay? Ayan, so ganun. And the other type is, hindi included dito yung uncompetitive, is yun yung pagka-pasok mo doon sa may active site, tsaka siya atake patalikod. So wala akong illustrations dito, guys, and I'm so sorry for that. And searchable naman siya sa may Google. At pala sinerch ko na. Oo. Sige. Basta ganun. Ayan, so these are here. binds active site, non-competitive binds other than the binding site. So these are the type of inhibition that you will produce. So punta na tayo dun sa may enzyme activity and regulations. So what is enzyme activity and regulation? Yung enzymes kasi natin participates in a metabolic process, specifically sa ating metabolic process ng ating katawan. Pero di ba, lahat naman tayo sa enzymes katawan natin is tayo when we are on a fight and flight response, syempre, di ba? Yan, we are so active. So the enzymes are working faster, stimulated. Sa enzyme activity regulation kasi, it's either it can be stimulated or inhibited. Okay. And those are, di ko natin, ang types of regulation of enzyme activity na madidiscuss sa ating next lecture is the covalent modification, such as the phosphorylation of hydroxyl group, attachment of adenosine monophosphate to a hydroxyl group, reduction of cysteine disulfide bonds, and then next naman is yung activation by proteolytic cleavage. Then third is the allosteric control. So yung enzymes natin, hindi lang siya basta-basta function ng function ng function. Meron din tayong regulation dyan, such as yung mga inhibitors natin kanina, is nakatulong din yan so that they can inhibit the function. Okay, may attach naman sa may ninyo, attach YouTube video in order for you to understand it better. So these are the multi-subunit model for the cooperativity of the positive and negative cooperativity of the regulation of enzymes activity, whereas the concerted model and sequential model. Ayan. These are also the regulatory subunits modulate the activity of the catalytic subunits of our enzymes. So that's it for our lecture for today, and I hope you get the big picture of our discussion. Ayan, so nakompleto na natin. And I think and I hope pala na you can recall all the topics and the principles that I discussed in this lecture using this concept map that I made. So thank you for listening, guys. And that ends our di pa pala tapos. Sorry. Okay? Punta na tayo dun sa may enzyme kinetics. And sa enzyme kinetics na yan this is the mathematical model in order for us to compute the numerical value of the enzyme kinetics or yung rate niya, let's say in terms of concentration per second. Basta concentration per unit time. So ito yung niya. These are the simultaneous derivation, also known as the Michaelis-Menten equation, ayan, or the Km, widely varies among different enzymes. So nagbabari yan. Depende sa enzymes na ginagamit nyo. The Michaelis-Menten equation. In order for us to calculate the velocity or the enzymes natin for the reaction progress of the reaction para ma-calculate natin yung numerical value. So ang mga variables diyan is yung ating the Vmax or the velocity max, the substrate concentration, and the Michaelis-Menten constant. Ayan. So these are the natin. So the Km, i-discuss naman yung totally sa next lecture. Ayan. So may specific value siya. Ayan. So for the double reciprocal plot, if you wish to do a linear regression analysis for you to calculate the enzyme or the Michaelis-Menten constant. So ayan yung gagamitin nyo. Yeah. So the Line-Weaver-Burke plot for the slope is you can calculate the Km over Vmax, 1 over V sub O, or the velocity extrapolated. Ayan. So that's it for discussion for today, and I hope you get the big picture. And ang pinakamahirap lang naman sa may part na ito, guys, is yung ating enzyme kinetics because it involves the calculations for the numerical value for the activity of our enzymes. Hope you learn from this topic. And sana mas naiintindihan ninyo mas maigi kung ano bang function ng enzymes. The further discussion of the enzymes, their purpose will be discussed on the metabolism. Mas ma-appreciate nyo sila sa discussion natin doon sa metabolism. Thank you for listening, guys, and goodbye.